so as a reminder, we have the next 3 Fridays, including this one, are heart and vascular focused in nature, including next week, which will be in the Graham Center Ballroom. So we will not have seminar in this room next week. It'll be over on main campus for our Heart Day Symposium. So if you're looking for it next week, join us in Graham Center Ballrooms. Also, next week, there's a ton of cardiovascular activities all throughout the week. So make sure you're on the lookout for those. We posted, emailed around on Instagram. Follow Healthy Hearts Club that has a lot of information about some of those outreach activities. So to kick off Heart Month, we have an outstanding lecturer who I'm super excited to welcome here today, Dr. Chiara Bellini, who is Associate Professor and Associate Chair of Bioengineering at Northeastern University in Boston. She completed her Ph.D. in Biomedical Engineering at the University of Calgary after completing both her master's and undergraduate work in Biomedical Engineering in Milan, Italy. She and I first met at an important research conference, I believe, last year on vascular mechanics and mechanobiology. And I was really excited to learn about her research and then subsequently invite her here with us. So her research includes both experimental and computational cardiovascular biomechanics, a lot of vascular biology, and a whole stream on how environmental stressors influence cardiovascular aging and remodeling as well. Her work has been supported by private foundations and federal funding, which includes significant NIH funding. She's active in both the American Society of Mechanical Engineers and BMES, and she served as the co-chair last year of the BMES meeting in San Diego. So for those of you who got to attend that, it's a fantastic meeting. She's to thank for that. And she is also extremely passionate about training the next generation of interdisciplinary scientists and heads the Ph.D. program at Northeastern University. So we are super excited to have her with us today.
Good morning, everybody. Thank you for the introduction and thank you for the invitation. It is really a pleasure to be here. As you probably guessed from the title of my talk, my lab is broadly interested in cardiovascular health. Cardiovascular disease remains the leading cause of death in adults 65 and older, and conversely, older age is a risk factor for the development of cardiovascular disease. Metrics of cardiovascular function have emerged as reliable indicator of cardiovascular age, which is often regarded as a surrogate for biological age. Amongst those, pulse width velocity, which you see there in the plot, is a spatially average measure of aortic stiffness and doubles between 20 and 80 years of age. Stiffening of central arteries increases the workload on the heart and propagates the pulse wave towards the microvasculature, possibly causing an organ damage. While aortic stiffening naturally occurs with age, environmental factors and stressor as well as lifestyle choices may accelerate this process, causing premature aging. And so in the map on the United States up there, each state is color-coded in a different shade of red that is proportional to the percentage of adults whose heart rate exceeds their chronological age by at least 5 years. Amongst these environmental stressor is inhalation of environmental pollutants, including cigarette smoke. Although this is not necessarily pertinent to the vasculature, I'm spotlighting here a paper that was published 10 to 15 years ago where they compared a smoking twin with a non-smoking twin to show the cigarette smoking accelerate the aging of the skin. And this is really a stark reminder how inhalation of these environmental pollutants may fast-track tissue aging. So within this broader context, my lab is interested in using engineering principle to predict the occurrence of and optimize the treatment for adverse cardiovascular events that occur because of environmental stressors that accelerate this aging process. So today I'm going to tell you 2 stories. One is on the mechanobiology of aging for the aorta, the main vessels in our body, and the other one is the effect of smoke inhalation, fire smoke inhalation on cardiovascular health. Both of these studies require the characterization of the passive mechanical response and vasomotor function of mouse arteries. They're both preclinical studies. So to do so, we build that system. It's a custom pressure inflation system where we secure a aortic sample in between these two glass cannulae up here, and then we can control the axial stretch as well as the luminal pressure, which essentially controls the circumferential stretch. At the same time, we can measure force through that load cells, and then there is a camera in the back that's not shown there that measures the outer diameter. So using this device, we can perform what we call inflation extension test, and so we keep the artery at in vivo length, or plus or minus 5% of that length, and then we vary pressure between 10 and 140 millimeters of mercury and measure outer diameter. At the same time, we can keep the luminal pressure set and then change the axial stretch until the axial force reaches a set value. We use this data to inform a semi-inverse constitutive model to determine the constitutive coefficient of this relationship, which then we use in a forward model to estimate metrics of interest under in vivo loads, including stress, stiffness, and energy. All these words you're going to hear through the presentation, which really represents the passive response of microstructural proteins within the aortic wall, including collagen, which is mainly in the outer portion of the vasculature, and elastin, which is in the medial layer close to the smooth muscle cells. We can also use this system to perform vasomotor challenges, where what this represents is an artery that is pre-constricted with phenylephrine, and then we provide increasing doses of acetylcholine to evaluate the recovery in outer diameter. And what that does is that it let us know what the signaling between the endothelial cells that line the luminal surface of the wall is with, and their communication with the smooth muscle cells which are in the central part of the vessel wall. So within this methodological frameworks, I'm just now going to discuss our work on aging. That was in collaboration with Bamar and Kellawans at NYU. While other groups have evaluated and compared cardiovascular function between young and aged mice, we set to determine the time course of these events throughout the mouse lifespan. So we chose our endpoint based on the survival curve of female wild-type mice, which we built out of a large database from the Jackson laboratory. And specifically, this 84-week endpoint, which is our aged endpoint, we chose it to be before the rapid decline that occurs past 100 weeks of age, which it's more representative of end organ failure rather than aging per se. We wanted to evaluate whether the different segments of the aorta age synchronously. And so we performed, using that system that I showed you, inflation extension testing on the ascending and descending thoracic aorta, as well as the suprarenal and infrarenal infrarenal abdominal aorta. Just as a reference, the heart is right here. What we found is that if we compare the cyclic distensibility between diastole and systole, so throughout how the aorta essentially deforms throughout the cardiac cycle, the ascending thoracic aorta closest to the heart experienced the largest decline in cyclic distensibility, which is still significant but muted in the descending regions, but then completely resolves, at least by that age, in the abdominal aorta. So for this first part of the talk, I'm just going to focus on the ascending aorta alone. To unbiasedly catalog the multicellular transcriptonic alterations that occur during aging, we perform single-cell RNA-seq on ascending thoracic aortic tissues from 12 and 84-week-old mice using the TNX genome platform. Unsupervised clustering identified 12 cell populations based on the most expressed genes, and then we use canonical marker to determine sets of vascular cells, specifically endothelial cells, vascular smooth muscle cells, and fibroblasts, as well as immune cells, specifically T cells and macrophages. We also performed flow cytometry in order to evaluate how cell wall proportions change with age, and that told us that there is a twofold increase in both T cells and endothelial cells proportion in age compared to young tissues, while vascular smooth muscle cells decline and the macrophage proportion remains the same. As a next step, we performed gene ontology analysis to identify signaling pathways that were altered with aging, and we found enrichment for signaling pathways related to senescence, ECM remodeling, and acquired immune responses in age compared to young tissues, with a decline in pathways related to autophagy, endothelial cells integrity, as well as angiogenesis. Because T cell receptor signaling was one of the most abundant in aged tissues, we further dissected the nature of T cell distributions within aortic tissues of young and aged mice. So our initial analysis identified two sets of CD3 positive T lymphocytes in aortic tissues. And by immunofluorescence, we found that these were mostly localized in the adventitial layer, so the outer layer of the aortic wall. In the single cell data set, the CD3 positive T cells were enriched for two genes, RAG1 and LCK. RAG1 provides instruction for one of the fundamental components that helps with T cells and B cells maturation and development, while LCK is involved in T cell development and T cell receptor signaling. We then took these two cell population of CD3 positive T lymphocytes, and we used unsupervised clustering to further distribute them, and we found 5 different clusters. All of them increased and expanded with age, but particularly this cluster 0 and cluster 1. And this cluster 0 is where that RAG1 and LCK were the most abundant. We then moved on to ECM remodeling and focused on vascular smooth muscle cells in the media. We found 2 populations of vascular smooth muscle cells. The first population was enriched for genes that are associated with proliferation and adhesion, and that population decreased with age, suggesting that this function may also be affected with aging. The second population had more of a proliferate phenotypes and was preserved during aging. However, we found a decline in several markers that are associated with ECM upkeep and remodeling, including elastin, decorin, biglycan, and tissue inhibitor or matrix metalloproteinases 2 and 3. Moving on to adventitial fibroblasts, perhaps not surprisingly, we observe a upregulation of several genes in the collagen superfamily. To corroborate these transcriptional inferences, we then quantified the microstructural composition of the aortic wall using Mason spectrum stains. What you see there in purple is smooth muscle cell cytoplasms and blue is collagen. So we quantified the amount of collagen and found an increase in the area fraction of collagen both in the media and the adventitia. And in both cases, there was a significant positive correlation between the increasing age and the collagen area fraction. This collagen-driven remodeling supported a rightward shift in the pressure diameter response towards larger diameter. Progressive wall thickening and circumferential tissue stiffening contributed to decrease the cyclic distensibility between diastole and systole by a factor of 2.4 between 12 and 84 weeks of age. Likewise, a progressive decline in axial stretch contributed to decrease the elastically stored energy between young and aged tissue, and this is responsible for blood flow augmentation. So overall, these data suggest that the structural stiffening of the aorta occurs progressively with age and impairs the ability of the aortic tissue to descend and recoil throughout the cardiac cycle under pulsatile loads, which of course contribute to the functional decline of the aorta with age. Because we observed a region-dependent remodeling of aortic tissue, we then we build a bilayer model of the aortic wall where we calculated or predicted the transmural distribution of circumferential tissue stiffness within young and aged aortic tissues in blue and red, respectively. To orient you on these plots, the luminal side is on the left, the perivascular side is on the right, so this is the side that is in contact with the blood. And then that vertical line indicates the border between the media and the adventitia, which changes with age because the outer layer of the wall, due to the collagen deposition, occupies a larger fraction of the wall in aged tissue. So these analyses show that stiffening of aortic tissues occurs not only in the adventitia layer, as is accepted and expected, but also in the media layer. Immunofluorescence also indicated accumulation of these cytoskeleton cross-linker smooth muscle alpha actinin 2 in the aortic media of aged tissues. And so together, this evidence indicates that there may be a co-regulatory mechanisms of extracellular matrix and vascular smooth muscle cell stiffening with aging through collagen deposition and cross-linking of the actin cytoskeleton. So recent evidence has shown that embedding smooth muscle cells in a stiffer matrix boosts their calcium signaling as well as their ability to contract. And so based on this evidence, we first confirmed that transcripts, as shown there, and expression within the tissue via immunohistochemistry, which is not in the slide, of myosin heavy chain 11, MYH11, and smooth muscle alpha actin 2, or ACTA2, were preserved with aging, indicating that the contractile machinery in vascular smooth muscle cells remain intact, and then suggesting that smooth muscle cells may retain their contractile phenotype with aging. We also observed an age-dependent increase in vasoconstriction in response to KCL of ascending aortic tissues under circumferentially isobaric and axially isometric conditions. So we kept constant pressure and constant axial length that represents in vivo loads. And so really this led us to look into voltage dependent and calcium permeable ion channel, particularly piezo-1. Why piezo-1? First of all, upregulation of piezo-1 has been shown in several cell types in conjunction with matrix stiffening. And second, our collaborator has shown that alpha-actinin 2 mediated activation of piezo-1 modulates vascular smooth muscle cells behavior in stiffer aneurysmal tissues. So we stayed for piezo-1 and found increased expression of piezo-1 in the aortic media in aged tissue compared to young tissues. And these colocalized with the MYH11 signal, suggesting that piezo-1 was expressed by smooth muscle cells. And so these led to two questions. So the first one is whether piezo-1 activation contributed to the enhanced vasoconstriction that we observed in aged tissue compared to young tissues. And second one, whether it contributed to the maladaptive remodeling of aortic tissue that decreased vascular function with age. To address the first question, we first confirmed that activation of piezo-1 by IODA1 recapitulated the enhanced vasoconstriction in young tissues that we have observed in aged tissues. So these really convinced us that we should spend the time and money to develop a conditional knockout mouse model where we deleted piezo-1 in smooth muscle cells. And so we did that, and I'm gonna refer to that mouse as PSO1 knockout mouse. Opposite to the age-dependent behavior that we observed in the wild-type aorta, the maximum vasoconstriction to KCL was preserved in, or was comparable in young and aged aortic tissue from PSO1 knockout mice. And so that addressed the first question. To evaluate how activation of PSO1 or whether activation of PSO1 contributes to the change in mechanical properties, passive mechanical properties of aortic tissues, we then perform inflation extension tests in PSO1 knockout aortic samples. Mirroring the wild-type behavior, we also saw a widening of the lumen with age, which is controlled by endothelial cells, and we really didn't do anything to the endothelial cells. However, loss of PSO1 in vascular smooth muscle cells preserved tissue thickness and prevented circumferential tissue stiffening, which decreased by half the decline in cyclic distensibility of aortic tissues. Likewise, there was no differing axial stretch and elastic energy storage between young and aged samples from PSO1 knockout mice. So taken together, these data suggest that loss of PSO1 rescues, at least in part, the functional decline that occurs with aging in the mouse ascending thoracic aorta. So in summary, this study identified intrinsic vascular maladaptation that occurs with aging in ascending aortic tissues, and also identified PSO1 as a gyrogenic signal that is, at least in part, responsible for changes in the function and structure of the aorta with aging. Now, before moving on to the second part of the talk, I just want to take a couple of minutes to discuss where we're going with this and what is the next step in this space. So all the data that I showed you so far are from female mice. And mice unfortunately do not undergo natural menopause, but we all know that women are at a higher risk of developing cardiovascular or are lower risk of developing cardiovascular disease ahead of menopause, but then their cardiovascular risk rises to match that of men after menopause. And recent evidence has shown that that may have something to do with an accelerated pace at which aortic stiffening occurs in women after menopause. So we developed this mouse model of menopause using a ovotoxin called BCD. And just some preliminary data here showing that if we focus on the perimenopausal window, we found an increase in stiffness, in aortic stiffness in mice that were treated with BCD, but not in those sham controls, suggesting that, again, the change in hormonal changes during menopause may work synergistically with aging to increase aortic stiffness. We also found that the menopausal transition impairs the endothelium-dependent vasodilation of the aorta, so vasomotor function of the aorta, which is another hallmark of aging, and we're going to talk about this in the second part of the talk. All right. So now that we have discussed how the aorta ages naturally, I'm going to move on to the cardiovascular effects of smoke inhalation. We have all heard in the news the intensity, frequency of wildfires have increased over the past few years, and while the fires themselves may be geographically contained, the smoke can travel distances away, and as a result, historical data from the EPA indicates a rapid increase in the contribution of particulate matter, or PM2.5, from wildfire smoke to air pollution. Inhalation of particulate matter, but also toxic gases in the smoke, has the detrimental effect to human health, and particularly cardiovascular health, through several mechanisms, and we're going to look at the systemic release of signal mediators in this. Of course, this is concerning for community exposure, and a student of mine a couple of years ago took a deep dive into the literature and came up with these association matrices where, again, each tile has a shade of red that depends on the number of publications that show a positive association with either components of the smoke, you have PM2.5, PM10, black carbon, carbon monoxide, and so on and so forth, or the day since the fire onset on the x-axis, and then on the y-axis we have cardiovascular ailments that are cataloged based on codes for insurance codes for ER visits or admissions to the hospital. But it is even more concerning for occupational exposure, and particularly wildland firefighters who experience high intensity exposure due to their proximity to the fire, but also their lack of use of personal protective equipment and the long work hours. Small but growing epidemiological evidence on wildland firefighter indicates that they are at increased risk of developing hypertension compared to the regular population. And so these really frame our research question: how does wildfire smoke inhalation increases blood pressure? And there are several ways that we could tackle these issues. We leverage our expertise in mouse model of inhaled pollutants to develop a control wildfire smoke exposure in mice that mimics the occupational exposure in wildland firefighters. To do so, we partnered with Michael Gorn at UC Berkeley to develop this system, this furnace that generates lab-scale fire smoke that is representative of wildland fire smoke. And this system has a heater that's mounted on a stepper motor and that travels on a quartz tube that has inside a quartz bowl. In the quartz bowl, we put the fuel. Now, we can use different fuel. I'm going to talk about smoke from Douglas fir needles that's representative of the Midwest in this presentation, but we also looked at manzanita and eucalyptus that are more representative of the Southern California area. We can also control the combustion condition. I'm going to talk about smoldering here, but we can also do flaming. And then we can dilute the smoke and pipe it to mice in an exposure chamber. So now, using this system, we can build these calibration curves where we can predict the PM concentration, particulate matter concentration in the smoke, the carbon monoxide concentration in the smoke, and other gases depending on the fuel mass that we put on the quartz boat. And we can also evaluate the percentage of carboxylated hemoglobin in the mouse blood. And the goal of these first study was to maximize the particulate matter in the smoke so that the COHB level, carboxyhemoglobin level, remain below injury threshold, which is approximately 15%. And so we settled to expose mice to smoke with a particulate matter concentration of 40 milligrams per liter cube. Of course, any preclinical model is, or the significance of any preclinical model depends on their ability to actually mimic human conditions. And so a talented student of mine who is now a postdoc at the University of Michigan came up with this exposure equivalency where he matched the cumulative deposited mass between mice and wildland firefighter normalized by the surface area of the lung in the two species. In order to do so, he calculated the deposited mass per exposure hour, which is the product of the particulate matter. That's the 40 milligrams per meter cube that we discussed in the previous slide for mice, and then for wildland firefighter, we just use average data from field measurements. That minimum volume, that is the product of the respiratory rate by the tidal volume, which is how much air we inhale. And for mice, we perform this measurement called plethysmography. And I just want to point out, this is a breathing waveform, I just want to point out to this long pause during expiration. And that is a typical response of a mouse or a rodent in general when they inhaled a irritant substance. And then for firefighters, we calculated the minimum volume for each fraction of their shift, depending on their heart rate, because that's easy to measure during firefighter operation. And then finally, we calculated the deposition fraction, and to do so, we partnered with Jessica Oakes, a colleague of mine at Northeastern, And we use a 1D finite element model to solve a Vection diffusion equation and calculate the percentage of the inhaled particle that then deposit and stick to the surface of the lungs by diffusion, impaction, or sedimentation. So once we calculated this deposited mass per exposure hour, we then could plug it into our equivalency analysis, account for the surface area of the lungs, because of course it's different between mice and firefighters. And we came up with this equivalency where 6,500 hours of exposure in firefighters, which on average correspond to a 7-year career, correspond to 80 exposure hours in the mouse, that 2 hours a day, 5 days a week, end up being 8 weeks of exposure. So another student of mine, Jackie Matz, who is now a postdoc at Tufts, did exactly that. So exposed her mice to smoke for 8 weeks, and then she also doubled it to 16 weeks. And because of the linearity within the equivalency analysis, that essentially corresponds to a 14 years firefighting career. And that's the data that I'm going to show today. And she collected some data along the way, but then at the end, used the system that we discussed to characterize the behavior of aortic tissue, both actively and passively. So as I mentioned earlier, one of the mechanisms by which inhaled substances can cause extrapulmonary effects, including cardiovascular effects, is through the release of signaling mediators within circulation. And we first look at pro-inflammatory cytokines because of their established association with cardiovascular disease, and also because of their involvement with fire smoke inhalation. So we measure the plasma concentration of TNF-alpha, IL-1 beta, and IL-6 throughout the exposure duration. And this is baseline measurement, meaning that they were taken before the daily exposure of mice. So we observed a transient increase in TNF-alpha within the first week of exposure, then returned to baseline. But by week 12, both TNF-alpha and IL-1 beta were consistently elevated in mice. While IL-6 was only elevated at the end of the exposure, which makes sense because it's downstream signaling, signaling-wise to the other two cytokines. So now, endothelial cells that line the surface, the luminal surface of the vasculature are in direct contact with circulating blood, where all these signaling mediators are, and therefore there represents a critical interface for the interaction between vascular tissues and these pro-inflammatory cytokines. Endothelial cells constitutively express membrane receptors for IL-1 beta and TNF-alpha, which confirm direct responsiveness to these cytokines. They do not express the receptor for IL-6, however, they can still interact with IL-6 via trans signaling. Therefore, the induction of pro-inflammatory cytokines by fire smoke inhalation is mechanistically poised to elicit endothelium-dependent vascular responses. IL-1 beta and TNF-alpha converge into NF-kappa B signalings. Therefore, we consider NF-kappa B as a central node for integration of vascular responses to fire smoke inhalation. We calculated the nuclear to cytoplasmic ratio of the for the localization of the P65 subunit of NF-kappa B, and showed that it was elevated in the endothelium of the aorta from mice exposed to fire smoke, which we expected. However, we also saw an increased nuclear translocation of NF-kappa B in medial smooth muscle cells and adventitial fibroblasts, which suggested that the pro-inflammatory or the inflammatory signal had traveled through the wall. Because NF-kappa B is a transcription factor that promotes the transcription and then expression of additional pro-inflammatory cytokines, we also evaluated the tissue distribution of TNF-alpha and 1-beta, IL-1-beta, which was elevated, mature TNF-alpha and IL-1-beta, which was upregulated in both cases in tissues from mice exposed to virus. Okay, so this really identifies a first amplification loop of vascular inflammation where pro-inflammatory cytokine-mediated NF-kappa B activation promotes the release of additional cytokines that can, through paracrine and autocrine signals, can then propagate the inflammatory signals through the aortic wall. NF-kappa B also upregulates the transcription and expression of the catalytic subunit 2 or NOX2 of an enzyme, NADPH oxidases, in both endothelial cells and fibroblasts. Most cells don't have NOX2. And immunofluorescence confirm a increased NOX2 signal in both the intima and the adventitial layers of aortic tissues from mice exposed to virus smoke. NOX2 is the primary source of superoxide in the vasculature, and upregulation or overexpression of NOX2 in endothelial cells increases vascular superoxide in epoenol mice, which are the mice that we're using for these studies. In addition to changes in NOX expression, the production of superoxide is also regulated by post-translational activation of the NOX enzyme. And it just so happens that pro-inflammatory cytokine can act as potent stimuli to activate NADPH oxidases and therefore increase the production of reactive oxygen species in the vasculature. It should be noted that NOX-produced superoxide maintains the ability to accumulate intracellularly in vascular cells, and that intracellular superoxide can trigger cytosolic signaling pathways. So based on this evidence, we evaluated whether fire smoke inhalation increased the oxidative burden in the mouse aorta. And to do so, we used this DHE staining. DHE is a fluorescent dye that is cell membrane permeable and that emits red fluorescence where it's oxidized. It is orange here, but just for visualization purposes. And so we observed an increase in DHE fluorescence in the aortic tissues of mice exposed to fire smoke. And again, it was through the wall. It was not localized only to the epithelium, which suggests that exposure to fire smoke increased the oxidative burden in the mouse aorta. We also treated aortic sample with this superoxide dismutase mimetic MMT BAP ahead of the DHE assay, which reduced DHE fluorescence, and this really acted as a negative control, but also confirmed the specificity of the DHE assay for superoxide. In addition to NOXes, uncoupled enos is another source of superoxide in vascular inflammation. And enos, or endothelial nitric oxide synthase, is constitutively expressed by endothelial cells, and it really produces the potent vasodilator nitric oxide. However, superoxide can react with nitric oxides to form peroxynitrates, and peroxynitrates cause uncoupled enos, which essentially divert the enzyme from producing nitric oxides to actually producing more superoxide. To evaluate whether uncoupled enos contributed to the oxidative burden in the mouse aorta following fire smoke inhalation, we then pre-treated the aortic sample with a enos inhibitor L-name and found that that decreased DHE fluorescence, which again implicated uncoupled enos in the generation of oxidative stress in fire smoke inhalation. To gather further evidence of enos uncoupling, we then perform an indirect assessment of peroxynitrates activity by measuring 3-nitrotyrazines. So peroxynitrates can react with tyrosine residues on proteins to form 3-NT, which is essentially a stable end product of reacting nitrogen species and is a biomarker of protein modification by peroxynitrates. And by immunohistochemistry, we showed accumulation of 3NT positive proteins closer to the intima layer of the aorta in mice exposed to fire smoke. Importantly, reactive oxygen species can act as a second messenger species to further activate NF-κB to produce additional signals such as IL-6. Therefore, the superoxide and in general reactive oxygen species in the mouse vasculature following fire smoke inhalation can fuel a second amplification loop of vascular inflammation in the context of fire smoke exposure. Because of this, we then evaluated the distribution of IL-6 within aortic tissues, which was increased in aortic tissues after fire smoke exposure. IL-6 upregulates transforming growth factor beta-1 expression as well as in fibroblasts and activity in several cell types, which we confirmed in our tissues. TGF-beta-1 is a pro-fibrotic cytokine that is capable of promoting ECM remodeling as well as collagen synthesis. And that happens through several mechanisms, one of which is the transition of fibroblasts into a myofibroblast phenotype with enhanced collagen synthesis capability. Consistent with this mechanism, we found that the expression of smooth muscle alpha-actin 2 or ACTA2 was elevated in the adventitial layer of aortic tissues exposed to fire smoke, and this ACTA2 is again a marker of fibroblast to myofibroblast transition. Given this molecular evidence, we then quantified the microstructural composition of the aortic wall and found an increased collagen content in the aorta of mice exposed to fire smoke, which we measured through picoserothread staining and then confirmed with immunochemistry for collagen 1 and collagen. Because of this fibrotic remodeling of the aortic wall, this suggested that fire smoke inhalation may affect or may impair the ability of the aorta to act as a compliant pressure reservoir throughout the cardiac cycle, which would eventually increase our blood pressure. Again, this is the evidence that we were trying to explain. So by the end of the study, systolic pressure was slightly but significantly elevated in mice exposed to fire smoke. To accommodate this larger pressure load while maintaining normal physiological stress, mechanobiological target, often in vascular tissues, the vascular wall thickened and the mechanical response of aortic tissue expanded to the right towards larger stretch. These occurred at the expense of increased tissue stiffness, again that slope of that curve, and decreased elastic energy storage, the area under that curve. And we already discussed how these two metrics are functionally significant because cells are mechanosensitive to the substrate stiffness, and store energy really indicates the ability of aortic tissues to deform and recoil and augment diastolic blood flow. Consistently, we found a decrease in the cyclic distensibility of the aorta between diastole and systole in following fire smoke exposure. So going back full circle to the beginning of the talk, the distensibility is an inverse metric of structural stiffness, and we know that the structural stiffness of the aorta increases naturally with age, suggesting that fire smoke inhalation may promote a accelerated aging phenotype in the vasculature, which is consistent with an increase in blood pressure that has been measured in firefighting. So now, vascular smooth muscle cells retain their capacity to reorganize their actin cytoskeleton and enhance force production in response to matrix stiffening, which is what we measured, which eventually could further decline aortic compliance in vivo. However, the vasoconstriction in response to phenylephrine was comparable in the aorta of mice exposed to fire smoke compared to filtered air. Vascular smooth muscle cells are effector vascular tone, and that does not only mean contractility but also mean dilation. And so that always often implicates the nitric oxide cyclic GMP signaling cascade. And so to evaluate whether fire smoke affected that signaling pathway, we performed we probed the endothelium independent vasodilation of the aorta, where we provided a nitric oxide donor called sodium nitroprusside, and again found no difference in two exposure groups. So that evidence combined indicates that fire smoke exposure does not or preserves muscle cells contractility as well as their response to nitric oxide. However, accumulation of 3NT positive proteins within the aortic endothelium, as well as, or nearby the aortic endothelium, as well as evidence of enos uncoupling, suggests that there may be a decrease in nitric oxide bioavailability, endogenous nitric oxide availability, so that one produced by enos, following fire smoke exposure. So based on this evidence, we then probed the endothelium-dependent vasodilation in response to increased doses of acetylcholine. And we found that fire smoke inhalation prevented the recovery of baseline diameter in pre-constricted aortic sample. As you can see, in aortic sample exposed to fire smoke, they were pre-constricted and provided acetylcholine to vasodilate. They don't recover 100% diameter, not even after the larger acetylcholine dose. And finally, to evaluate how much of this is due to the decreased nitric oxide production by enos, we pre-treated the aortic sample with the enos inhibitor L-name ahead of the acetylcholine challenge, and then repeated the acetylcholine challenge. As you can see, this treatment further attenuated the response to acetylcholine in aortic samples that were exposed to air, but not in those that were exposed to fire smoke. And so this suggests that fire smoke may have already removed that response. And so taken together, this evidence indicates that fire smoke inhalation impairs the endothelium-dependent vasodilation through abrogation of the nitric oxide response. This is expected to increase peripheral resistances and then diastolic blood pressure, again, consistent with the epidemiological evidence found in Wallace firefighters. So in summary, this study so far showed that inhalation of fire smoke at occupationally relevant concentrations causes aortic stiffening as well as increased peripheral resistances, which both can contribute to alter blood pressure and increase the cardiovascular risk. So now, where are we taking this? This preclinical study was really helpful because it let us know what we have to measure in people. And so we're now doing that. We have an IRB-approved protocol for taking measurements in Wallace firefighter. As a matter of fact, I spent the first part of the week in Tallahassee, where I was working with the US firefighters in the USDA to collect measurements. This is one of my students, Victoria, who is leading this project. This is not in Tallahassee, this is in Georgetown, California. We were at a training camp for prescribed burn, and so she was learning how to use the drip torch. But most importantly, she collected this data where it's the equivalent of the vasomotor function that we tested. We did in the mice, but she used a laser Doppler methods to look at vasomotor function on the blood vessels of the skin in Wallace firefighters. We're also collecting PBNC, so peripheral blood mononuclear cells, to look for some biomarkers of exposures there. Okay, so before concluding, I just want to bring up one more time the research roadmap of my lab. There are lots of projects that I did not have the chance to talk about today. I'm happy to do so after and in the meetings that we'll have through the day. But I hope that I gave you a flavor of what my lab is interested in. And so I'm going to stop here, acknowledging all the students that worked on this project, the funding agency, and thank you for your support, for your attention. I'll be happy to answer any questions you have.
see you. All right. Thank you so much for sharing all of that with us. I really like your presentation, Tom, and your visuals are awesome. My question is a little more on the philosophical side. That's who I am.
I think they were expecting it, that's what we like.
Yeah. So the association between stiffness of the vasculature due to some sort of negative stimuli like smoke inhalation makes perfect sense. Right? How that there may be a causal relationship between vessel stiffness and induced hypertension, right? But then on the other hand, you're saying that vascular stiffness in aging is natural, like that happens. Is that some sort of compensatory mechanism that mammals have to compensate for loss of cardiac function or what have you in nature? Like, I guess my question is, does that vascular stiffness always lead to adverse outcomes, or are